Hey, this is William, and I'll be talking about gene editing to treat atherosclerotic cardiovascular disease. What is it? It's a chronic, lifelong disease characterized by the buildup of plaque inside arteries. Plaque is made up of fats, cholesterol, and other substances. It narrows arteries, blocks blood flow, and can cause blood clots. The buildup is due to a chronic, slowly evolving vascular wall inflammation and lipid accumulation. It can lead to adverse cardiovascular events such as heart attacks, strokes, or even death. Risk of these adverse cardiovascular events increase when low-density lipoprotein levels in the blood are elevated. How do we treat atherosclerotic cardiovascular disease? We can treat it by lowering LDL-C levels using CRISPR-Cas9 in vivo gene editing. But first, we need to understand how LDL-C levels are regulated. The liver secretes PCSK9, which is a protease that binds to low-density lipoprotein receptors, also known as LDLR. LDL receptors are involved in removing cholesterol from the blood. PCSK9 prevents recycling of these LDLR receptors and increases its degradation. Therefore, it reduces the body's ability to lower LDL-C levels in the blood. In a normal person, LDLR is being recycled back to the plasma membrane, allowing it to remove more LDL-C. CRISPR-Cas9 in vivo gene editing for lipid-lowering therapy. Lipid nanoparticle delivery. Lipid nanoparticles are delivered by a single IV administration. Nanoparticles contain Cas9 mRNA encoding an adenine base editor and a PCSK9 specific guide. Nanoparticles enter hepatocyte liver cells through endocytosis and endosome formation. Breakdown of lipid nanoparticles and endosomal membrane releases active components into the cytoplasm. Cas9 mRNA is translated in the cytoplasm by a ribosome and combines with the gRNA to form a ribonucleoprotein. The ribonucleoprotein enters the nucleus to induce gene editing. It introduces a nonsense mutation in the PCSK9 gene by converting a single adenine thymine to a guanine cytosine nucleotide base pair. A nonsense mutation is a genetic alteration that causes premature termination of a protein during translation. Altered proteins become partially or completely inactivated. Basically, it turns a codon into a stop codon. Pre-treatment. PCSK9 normally binds to the LDLR, leading to degradation and increased LDL-C levels. Post-treatment. Nonsense mutation reduces PCSK9 gene levels. Inhibition of PCSK9 prevents degradation of LDLR, allowing it to recycle back to the plasma membrane. Treatment results in reduced circulating LDL-C levels. These are my sources.